So, hello and welcome everyone. Good afternoon, good morning, wherever you are, to the kickoff session of our new varvis webinar series with the first session on liquid biopsy in breast cancer using NGS. So we have put together a very nice program for you today. Next slide. There it is. We have three experts invited here in our session, covering all aspects of liquid biopsy. The first session is on the first part is on the clinical application. The second is on the wetlab, a new library preparation kit from IDT. And the third part is on validation and a nice software solution called varvis. At the end, we have a Q&A session, so we make sure that we can get all your questions answered. This is a very nice opportunity with the three experts in-house to get all questions that you have, you can ask them, just to make sure that we have all on the same level here, how to ask questions, depending on the setup you're using. On desktop, you have a webinar control panel where the question tab is like hidden here that you can open by click, and then you can type in your questions here. When you're using a mobile device, you can enter questions using the question mark icon in the header of the application. Before we get started, just a few words who we are as we are hosting this this webinar today. So my name is Yvonne Kassmann. I'm from Limbus Medical Technologies, and Limbus is a software development company located in the north of Germany, as you see here, right at the Baltic Sea. We are a medical device manufacturer, which means that we develop and operate our products according to international medical device standards. And our product, varvis, is cloud-based and a clinical decision support system enabling analysis of all kinds of NGS data, ranging from panels to whole exome and to cancer diagnostics, including liquid biopsy. And this is what our session is today about. So we start with the clinical application of liquid biopsy in breast cancer, which is presented by Dr. Simon van Amen from Aachen. So I'm happy to introduce to you Simon van Amen, who is a biologist by training and completed his doctorate thesis in Cologne. Hello, Simon.
Hello.
After some years in doing postdoc research, he turned to clinical diagnostics in 2016, where he gained a lot of experience in NGS panel diagnostics and whole exome. Since 2019 now, he is focusing on oncology, and he has established liquid biopsy using NGS technologies in-house himself. And we're happy to hear how this went and to see some data. So I'm handing over the mouse to you. Yeah. You're welcome to start.
Okay, yeah, thank you very much for this kind introduction, and also for the invitation to talk about our approach, how we would like to use liquid biopsy to improve molecular diagnostics in breast cancer. Here we go. Yeah, we are a group practice for hematology and oncology and laboratory medicine in Aachen, based in Aachen. And we are focused on comprehensive care for breast cancer patients. In our molecular oncology lab, we monitor circulating tumor cells by imaging flow cytometry. And we have already established liquid biopsy by digital droplet PCR. And so this is the topic of this webinar today. We are establishing NGS-based liquid biopsy approach. This liquid biopsy project is supported by Metavis, which is a non-profit association which is dedicated to support projects for the early detection of cancer recurrence. Yeah, I'd like to start with a short introduction into liquid biopsy. So yeah, cell components of either a tumor tissue or healthy tissue is released into the bloodstream, and this is a natural process which is triggered by apoptosis, necrosis, or even by active secretion. There are several cell components which are released into the bloodstream. For example, cell-free RNA, circulating tumor cells, cell-free DNA, and exosomes. And we are interested in circulating tumor DNA, so small DNA molecules which originate from the tumor. Yeah, what are our goals with liquid biopsy? So the main goal for us is that we want to identify cancer recurrence as early as possible, and this especially in patients which are at a high risk. And there have been studies already which could show that with liquid biopsy, one is able to identify or to see cancer recurrence 6 months earlier than by other methods like imaging methods like computer tomography. Second, we want to individually select and adjust therapies. And third, we want to monitor the therapy response by tracking patient-specific mutations. Yeah, there are several advantages of liquid biopsy. First, it's non-invasive besides a blood collection, and sometimes there is no tumor biopsy available, or the site is just not accessible. Then one could use liquid biopsy, and also it has been shown that the heterogeneity of the tumor is even better reflected by liquid biopsy than just by a tumor biopsy. And also very important, we are able to take serial sample collections and time series. Yeah, and of course there's a big challenge. We are looking for very rare events in liquid biopsy, and yeah, we have to deal with the limit of detection, which is way below 1%. Yeah, I would like to continue with some slides about recent progress in diagnostics in breast cancer and using liquid biopsy, and also specific treatment options depending on the mutation or on the gene. Therefore, I would like to cite some data from the plasma match trial. The plasma match trial is a big trial which is still ongoing, I think, with more than 1,000 patients with advanced breast cancer. And for these patients, samples were collected and cell-free DNA was analyzed by a comprehensive NGS panel, the Guardant 360. And yeah, the striking finding was that they could identify a somatic alteration by liquid biopsy in 92.9% of the participants. And this is a great potential to identify a mutation which might be targetable by a drug already, or might become targetable by a new drug in the future. And this is also from the data from the plasma match trial. This chart shows the genes which are most frequently mutated in advanced breast cancer. The major genes are TP53, PIK3CA, and ESR1, and to a lesser extent GATA3, ARID1A, and ACT1. Yeah, and yeah, as an example, yeah, some examples. There are already some drugs on the market or in clinical trials which address specific genes. For example, mutations in PIK3CA, they are targeted by a drug called Piqray. The agent is called alpesilib. Then mutations in ESR1, they are known to cause resistance to aromatase inhibitors, which are commonly used in the treatment of breast cancer. And then we have two more ACT1 mutations can be targeted by cabivasertib, and mutations in HER2 by niraparib. And there are more trials going on, and there will be more drugs available which target specific genes and mutations. Yeah, I would like to, as an example, highlight one drug, which is a PIK3CA inhibitor, Piqray, which was recently got approval in the European Union, and the drug can be applied only if a mutation has been confirmed in the patient in PIK3CA, and this is the case in 40% of hormone receptor positive HER2 negative advanced breast cancer. So these 40% of patients are eligible for this kind of treatment. And yeah, mutations in PIK3CA, they cause a hyperactivation of the alpha subunit of PIK3 and hyperactivation of the ACT signaling pathway. And this promotes tumor growth and resistance to endocrine therapy. And yeah, the mode of action of this this new drug, Piqray, inhibits the alpha subunit of PIK3 and blocks the ACT signaling pathway. Yeah, and the efficacy of this drug has been studied in the SOLAR-1 study. In cohort 1, they investigated, the participants, they got treated with Piqray and fulvestrant. Fulvestrant is a hormone receptor antagonist, so this is used in combination. And in cohort 2, this was the control group, placebo, and fulvestrant. And they found out that the inhibition of PIK3 alpha subunit induces estrogen receptor transcription, and this means that the endocrine therapy might become effective again. And the outcome of this study was that in the Piqray fulvestrant cohort, the progression-free survival was increased by to 11 months compared to 5.7 months in the placebo group. Okay, this as an example. Now I would like to present our approach, how we want to use liquid biopsy in our clinic. So this is an overview of a typical current diagnostic procedure in our clinic. So if we have a tumor material available, then we send this for tumor sequencing to Foundation Medicine. And yeah, this is the next very comprehensive sequencing test of the tumor biopsy. Then we get the report, which might, for example, tell us that this patient's got a PIK3CA mutation in the tumor. Then we perform a follow-up care, so we see the patient like every 3 to 6 months again, and are able to again collect cell-free DNA sample. Yeah, and then the oncologist, if the oncologist suspects a mutation in PIK3CA or in ESR1, then we are able to perform ddPCR. And yeah, depending on the result, the oncologist can select the treatment or adjust the treatment. So this is yeah, the current state. The thing is that, of course, we at the moment were only able to look at hotspot mutations in these 2 genes, and yeah, we miss quite a lot, and we want to extend our analysis to further genes. From the tumor sequencing reports of our patients, we also we know which genes are most frequently mutated. And this is similar to what was seen in the plasma match trial. We have the main genes in which we find mutations are TP53 and PIK3CA, and yeah, to a lesser extent ESR1 and ACT1. And yeah, one gene TP53 is of special interest for us. TP53 is commonly mutated in triple-negative breast cancer, and triple-negative breast cancer is associated with a worse prognosis and also with less treatment options. And therefore, yeah, it's one aim to see TP53 mutations to be able to early detect cancer recurrence. Yeah, and corresponding to what we know from our patients, we decided to design a small NGS panel which fits to our needs, to our patients, with these 5 genes: PIK3CA, ESR1, ACT1, HER2, and TP53. And yeah, now the NGS-based workflow looks like this. So we collect cell-free DNA from the patient with a special collection tube which stabilizes the cell-free DNA. Then we use 2 centrifugation steps to avoid contamination with genomic DNA. And for the extraction, we use a Maxwell instrument from Promega. Then we perform quality control with a tape station to see how successful our extraction was. And then we come to the library prep. So we use IDT's XGen Prism kit for library preparation and capture. And the technique is based on a duplex sequencing, which allows us to detect very rare variants with a high precision, what we need in liquid biopsy. Then we perform sequencing on a mini-seq, and then we transfer the data to varvis. So we are a small lab, we don't have a bioinformatic unit. We send the data to varvis. They take care of the analysis and also support us with assay validation. And all our analyses are stored in a database online. And we also are able to track changes in the allele frequency over time. So this, of course, is also very important, as we want to monitor what happens. We don't have that many real results from patients yet, just a couple, but still I would like to show you one preliminary result as an example. So this is about a patient, 62 years old. She was diagnosed with breast cancer in 2016, and the breast cancer is hormone receptor positive, HER2 negative. In this case, we did not have sequencing data of the tumor, and the last ddPCR for PIK3CA was negative. So we used our new liquid biopsy NGS panel in August, and by duplex sequencing we could identify one suspicious variant in TP53 with a mutant allele frequency of 0.13%, which is very low, and of course this needs to be confirmed. But it's a suspicious variant, and bioinformatic tools predict this variant to be damaging, and also it's listed in ClinVar as likely pathogenic and in Cosmic in the Cosmic database as pathogenic. We need to repeat this, of course, but if we see this variant again in the next run, then we would have the possibility to follow this specific mutation in this patient by ddPCR. Yeah, thank you very much for your attention. And I also would like to thank Dr. Habetz, who's the PI of this liquid biopsy project. And I would like to say thank to my colleagues, Ilham and Sarah. They are responsible for this monitoring of the circulating tumor cells in our lab. And Mariam is our ddPCR expert. Thank you very much.
Okay, so thank you for this nice presentation. I would like to take the opportunity to ask the first question that was already available here in the chat. So the question is, how many hours or days does the workflow take from sample to result?
Okay, so I would say the library prep and the capture 1.5 days, and then the mini-seq runs of 24 hours approximately. So then the data transfer, I would say it's possible in 3 to 4 days.
Okay. So these were the questions so far. Maybe some more questions pop up. We will see you again in the question and answer session at the end. So stay tuned. So I would like to continue with our second talk. For which, oops. Yes, sorry. Slides are too fast and too slow at the same time. So the second presentation is about the XGen, the new XGen Prism library kit by IDT. And we have here as a presenter Peter Verhasselt. Welcome, Peter. Thank you for joining us here today. I would also like to introduce you to our guests here today. So yeah, there's a slide. Peter did his PhD in biotechnology and some postdoc in developing Sanger sequencing related technologies. As you can see, he has a long experience, more than 20 years in NGS and DNA sequencing and various applications, from whole genome sequencing to cancer diagnostics. He joined IDT in 2018 as the field application manager for NGS, and today he's here to present the new Prism kit. I'm handing over to you, Peter.
Okay, thank you for this introduction and good afternoon to the audience. So I will focus on part of what Simon has been presenting. So I will focus my talk a little bit on the Prism library prep kit, which has been developed by IDT, especially for quality compromised DNA samples, such as the circulating tumor DNA or the liquid biopsy and FFPE DNA. It seems like the slides are not moving under my control. Okay, so what I will present here today is first a very short introduction to integrated DNA technologies as a company, the products we have available for a next generation sequencing workflow, and some challenges that are usually encountered with FFPE and liquid biopsy samples when performing next generation sequencing experiments. Majority of the talk, however, will be focused on a technical presentation on the Prism library prep kit and some results that have been obtained internally. So integrated DNA technologies, or IDT, can really be considered one of the major oligosynthesis companies. We have a worldwide presentation with 5 production sites, 3 in the US, 1 each coast, and then also production in Coralville, where also the global headquarters are located. For Europe, we have a production site in Belgium, and for the Asia Pacific, we do have a production site in Singapore. On an average day, we will produce around about 90,000 oligos spread over something like 4,200 orders. Worth mentioning is that as of 2018, IDT has become part of the Danaher set of operational companies. Over the years, IDT also has built some product lines in which the oligos that we are making are playing a particular role. So for example, we have a PCR and qPCR product line where primers and probes are quite essential. In the functional genomics, we are using the RNAi oligos and guide RNAs together with the required enzymes. In synthetic biology, G-blocks and gene fragments are key players, and with the next generation sequencing applications, we are making use of oligos for generating adapters, blocking oligos, and probes that can do the capturing of the target DNA sequences. If we take a closer look on an NGS workflow, typically such a flow starts with extraction of the DNA or RNA out of the sample. Typically, the first step will be a library prep, from which you can directly go into sequencing for applications such as whole genome sequencing or methylation analysis. When focusing the sequencing technology more on the regions of interest, such as indicated by Simon with his example of 5 genes, typically people would do an enrichment. When we have a first library prep, then enrichment can be done with a hybridization with biotin-related probes. In an alternative approach, people can go directly from extraction to a finished library with a 2-step amplification system. And where IDT has products in, it's really a modular system. So for the library prep, we are offering the Lotus library prep for high quality DNA, but I will focus here more on the Prism for quality compromised DNA samples. With library prep also come adapters that have to be added for enabling sequencing on the Illumina platform. For the enrichment, as mentioned by Simon, people can order the capture probes also from IDT. We do have inventory panels, like for example, an exome for whole exome analysis. But we can also, as Simon has indicated, create a customer-specific panel for the genes of interest to the customer. And for those that are interested in PCR-based targeted sequencing, we do have our RAM-seq technology, but we will focus in this talk on the Prism. Historically, cancer biopsies have been collected and stored as FFPE samples, which present some challenges for analysis by NGS. So the DNA is quite often of poor quality and fragmented to a variable degree due to the fixation artifacts. Also, the quality control can be very complicated because due to internal damage, not all of the quantified material is fit for library prep. Biopsies can be very small, and the tumor content within those biopsies can also vary quite a bit. So typically such biopsies will contain tumor DNA next to the wild type stromal DNA. To come to the Prism library prep kit, this comes with quite some interesting features like a high conversion rate. The technology suppresses the formation of primer dimers and also will prevent more the production of chimeras. So in a classical library prep, there is the opportunity that chimeras are formed. This is the library fragments that ligate to each other, and by such create an artifact because those ligated fragments in the original samples were not next to each other. Adapter dimers can be formed. This is adapters that ligate to each other, and those will compete for library molecules on the sequencing. And the conversion rate is actually the fraction of the input molecules that end up in the final library. Why is this conversion rate so important? So ideally, as illustrated in the central part, ideally the molecules that you have in your sample, you want them all to be converted into the library, because this initial set of molecules will contain at low frequency the variants you're interested in. So with a high conversion rate library prep, majority of or a higher fraction of your library molecules will end up, of your original molecules will end up in the library, resulting in libraries with higher complexity, higher coverage, and you can find your mutations with a higher degree of certainty, so with more confidence and at a higher sensitivity. On the contrary, if you have a library prep kit with low conversion rate, only a minor fraction of your initial molecules might end up in your library, resulting in lower coverage, lower sensitivity, and by stochastic nature of how those molecules enter into your library, it might even happen that the variants you're looking for are not even part of the library you finally will be sequencing. So how does the Prism library prep kit works and addresses those problems? So the input for the Prism library prep kit is already fragmented DNA. So this can be naturally fragmented, like in the circulating tumor or in the liquid biopsy samples. For FFPE samples, if they are already dramatically degraded, they don't need an additional fragmentation. For higher quality FFPE DNA, we recommend at this point in time mechanical shearing as a first step. Mechanical shearing is the technology we have most experience with, but there is no indication that it should not work with enzymatically sheared DNA. So the input is fragmented DNA, and in a first step, the enzymatic reactions will happen to entry pair the insert molecules. So the entry pair will do a fill-in or a degradation such that the final molecules are blunt-ended. Then follows a two-step ligation, and these adapters used in the first ligation step have quite a specific structure. There are the longer molecules that are ligating onto the insert molecules. Those longer molecules do contain a molecular barcode. and they have specific modifications that are needed by the ligase that has been developed for this kit. The shorter molecules are having a blocking group at the 3' end. This prevents ligation to the 5' end of the insert molecules, but more importantly, the fact that the shorter molecules of the adapter are blocked for ligation, this prevents the adapter dimer from formation. We prevent chimeras to form because library or original DNA molecules that are in the library prep are not having the modifications that are required by the ligase. In the second step of the protocol, we will add a primer that is annealing to the stem structure of the longer molecule that is in the adapter. And this primer will displace the shorter molecule of the adapter and will displace it, remove it, and by such will also remove the blocking group, and the extended filled-in product can ligate to the original DNA molecules. This results in the bottom structure, which is a representation of a stubby adapter ligated onto the molecules of interest. Again, adapter dimers are not formed, and you can think of such a structure as the result of the ligation of the NEB adapters upon cutting open with the user enzyme. It's a similar structure. And finally, to label the samples with sample barcodes, there is a amplification reaction with unique dual index primers that are used to uniquely identify the samples. By using unique dual indexing, we are mitigating the index hopping problem, which can be observed with Illumina instruments that are using the patterned flow cells, like the NovaSeq, for example. A word about the molecular barcodes. Those are not random sequences. Those are really fixed sequences. It are molecular barcodes with a length of 8 nucleotides. And in the adapter pool, we are pooling 32 combinations. And since ligation events left and right of the insert molecule are independent from each other, this results in 1,000 combinations that are possible. It's 32 times 32. Because those UMIs have a specific sequence, we can discriminate PCR errors that would occur in the PCR steps downstream of the ligation. And by doing so, we can prevent the artificial inflation of the library complexity. So we have applied this internally to a few sample types. For example, first we have used the circulating fluid DNA. It's a reference sample where one genome has been mixed into the sample at a 0.25% frequency. Libraries have been made with a Prism library prep kit, and a capture reaction has been performed with a lockdown panel similar to the 5-gene panel that Simon has been describing, followed by data analysis. What we typically observe in comparison to other products on the market, starting from 10 nanogram of cfDNA, we are getting a higher yield. And also of note is that we are not observing the adapter peak that is visible in the central part of the fragment analysis patterns on the right-hand side of the screen. By also counting the start and stop positions of the insert molecules and taking into account the unique molecular indexes, we can calculate the number of unique molecules that are in the sample. And this is for the Prism library prep kit quite a bit higher than for the other two that have been tested. And on the right panel, you can observe that with increasing number of reads, the main target coverage is not getting to a plateau as early as with the alternative technologies, and that the mean target coverage is actually much higher. This is coming from a lower duplication rate, one end side, but also because with the molecular barcodes that are introduced during the library prep, we can include those in next to the start and stop positions to identify the unique molecules. Looking at the variants that have been called, we are looking here at sensitivity. So it's the frequency of the mutations or the fraction of mutations that we expect to find back to see which of those, which fraction do we really find in the data analysis. This is for the three technologies quite high, around 90% or above. The real benefit comes from the specificity, so the larger positive predictive value, because thanks to the molecular barcodes, and I think Ben will cover that more in his presentation, thanks to the molecular barcodes, we can perform error correction and remove majority of the false positives that have been called. In a final experiment, we've taken FFPE samples, reference FFPE samples with varying degree of degradation. So they're available in 3 categories. Again, performed library prep with a Prism, did a capture with the same lockdown panel, and performed data analysis. And what can be observed here is that with the Prism library prep kit on highly degraded DNA, we can get a library with a quality that the other technologies would only be able to get from high quality FFPE samples. When taking the severely degraded FFPE reference material, we made libraries starting from 3 different quantities of input DNA, and libraries have been made in triplicate. Then next, after the capture, we were then looking at clinically relevant variants that were present in those reference materials to see if we can find them back, and if we can find them back at the expected allele frequency. And this is clearly the case in all 3 replicates. We can find those relevant mutations, and those are in an expected allele frequency of 1 to 24. And also Simon has given the idea that 0.13% what is a result that can be achieved by the Prism together with the molecular barcodes included. So to wrap up, I think it's fair to say that the Prism library prep kit enables the low frequency variant detection within degraded samples, such as liquid biopsies and FFPE samples. This is achieved by the higher conversion rate that we can get as compared to the TA overhang ligation-based methods, and also because of the new ligase and the modified UMI-containing adapters that are used. It's definitely possible to find the variants at below 1% frequency, thanks to the use of the molecular barcodes in the library prep. With that, I'm going to open for questions.
Okay, thank you very much. Indeed, there was one question raised already. What is the lead time from ordering a custom kit to starting in the lab?
Okay, I think we have to discriminate between 2 parts of the complete workflow. There is a Prism library prep kit, which is available off the shelf. If we're talking about the panels that are used for doing the capture reaction, a lockdown panel such as Simon has used, that takes about 2 or 3 weeks from design to arriving in the lab for use.
Okay, very nice. So no more question at this point. Then I will switch to, thank you so far, Peter. We see you later.
Thank you. Thank you. See you later.
I'm trying to control so now it works. So the last part of our session today is about validation of liquid biopsy, knowing your analytical performance, presented by Dr. Ben Liesfeld from Limbus Medical Technology. Hi, Ben.
Hi.
I would like to introduce you as well to the audience. So Dr. Ben Liesfeld did his PhD in physics. From 2008 to 13, he was working in a startup for ophthalmological medical devices, where he was responsible for the development of computer-guided laser surgery devices. In 2013, then he moved to a clinical laboratory for genetic diagnostic, where he was establishing IT, software development, and bioinformatics department. So this was the first time you got in touch with genetic diagnostics. And there you learned about the huge potential and the challenges in NGS data analysis. So this led him to start his own enterprise, and he's a co-founder and managing director of Limbus Medical Technologies. And today he will present the product varvis and how this product is supporting, oh, now my slide doesn't work, supporting the validation of liquid biopsy.
Thank you, Yvonne, for the introduction. So let me start briefly with a short overview what varvis, our software varvis, actually does. It's a complete software solution for the entire analysis process of NGS data. It is a cloud-native medical device software, and that means that there's no investment required to start using the software, or to no investment in IT infrastructure. And we are able to deliver our services with equal performance across the globe in any country. And our software also provides immediate feedback about the quality and the analytical performance of an assay, and this is particularly important for applications like liquid biopsy. The software is suitable for small, from any scale, from small panels to whole genome sequencing, and it actually helps our users save costs in the laboratory because it makes some alternative methods obsolete, for example, by performing an automated CNV analysis. Well, how does the varvis software support liquid biopsy specifically? I'll walk you quickly through the workflow. We are starting with the raw data from the sequencing instrument. The varvis software performs the alignment. It also performs automatically the barcode processing, which may be quite a complex process depending on the technology that you're using. And it also performs variant calling in an automated fashion. And you can perform classification of variants, and you can create reports from those classifications, all in one seamless workflow. The onboarding process that we provide to our customers includes support and technology selection. So we have a good overview which technologies work well for specific applications. We also provide comprehensive training in our software, and as a special service, we provide the validation of the assays of our customers. And the validation is performed by comparing the output of the data analysis to the known and published reference data that when our customers are sequencing reference materials. And I think everyone who's performed validation of NGS assays knows how much work can be and how much effort can be involved in this kind of process. While the topic of my talk is targeting and knowing exactly your analytical performance, and there are two important parameters that you're looking at, and Peter already mentioned them. The first parameter is, of course, the sensitivity, and sensitivity in relation to liquid biopsy means, can you actually really preserve those few molecules that were present in your sample throughout the process, throughout the wet lab process, and make them visible in the results of your analysis? And the second parameter is typically called specificity, but in NGS we're rather using the term precision or positive predictive value, and that means, can you really distinguish, how sure are you when distinguishing between true and false? Can you tell apart a true variant from a false positive variant? And the degree of or the amount of false positives that you may be able to tolerate in your assay strongly depends on the existence of an independent method of confirmation. If there's no such method available, or if it's very expensive to confirm variants, then your assay must be designed in such a way that you have basically no false positives in your results. To just give you an overview how we differentiate liquid biopsy applications from other applications in next generation sequencing. Well, the application that probably most people are familiar with is germline analysis of human DNA, where you expect variants to occur at a fraction of more than 20% of your reads. You typically apply conventional deduplication. You basically remove duplicate reads from your data, and you operate at a sequencing depth between, let's say, 80 and 150x coverage. With, for example, when you're doing whole exome sequencing, and you have a very low duplicate rate in your data. If you are when you're trying to go to lower your limit of detection, if you want to detect lower frequency variants, you're moving to smaller regions of interest, you have to apply additional technologies like unique molecular identifier. You have to achieve a high raw coverage, and actually a duplication rate then is helping you. I'm going to detail how a duplication rate is becoming important in these kinds of assays. And kind of the utmost or the most difficult assay, the liquid biopsy, where you push the boundary of the limit of detection below 1%. This is where you need very complex technologies like duplex sequencing, very high coverage. And I'm going to show you how this process works, what the requirements are, and how you can actually validate that you achieve this kind of performance. So it seems, my list, it seems clear that it becomes increasingly difficult to go to a lower limit of detection. And let me quickly explain why that is. There's difficulty in detecting low or rare events in somatic or in liquid biopsy samples using NGS. And the reason for that is that there is noise in next generation sequencing. We talk about an error rate in our NGS data. With the error rate, we typically refer to the base calling error rate. If you're thinking of your sequencing 100 base pair long stretches of DNA with your instrument, and if you think of having a certain error rate in your instrument, the instrument is making mistakes by calling single bases, and if your read length is 100 base pairs and your error rate is about 1%, you can expect on average that one base in every read has a mistake, is false. Of course, you're repeating the sequencing, you're sampling this stretch of DNA many times at high depth. In each of those reads, there may be errors, some may be error-free. And if you're looking at a specific position now, a specific base that you are calling, and you may expect a number of false base calls at this position. And depending on your application, those errors may be important or not. If you're performing germline analysis, where you expect variants with a high frequency, then this is probably not important. But if you're going to if you lower your limit of detection, if you're going to a low limit of detection, then this becomes increasingly important. Because keep in mind, when you have a 100 kb NGS panel, this means you're independently interrogating 100,000 different genomic coordinates, and this means that you have 100,000 separate statistical tests. So these may be small probabilities to encounter an error at a specific position, they all add up. So across your entire assay, you may have a significant probability to have false positives across your entire assay. There is an interesting paper by Petrakova and Frontiers in Oncology from 2019, which details the statistical model behind that. So how there's noise in the NGS data, how can we deal with the noise? As Peter already mentioned, we can barcode our DNA molecules. So we start with a single molecule and we apply specific barcoding, and we barcode the molecule in such a way that we can differentiate between the top and the bottom strand of the DNA molecule. Then the top and bottom strand are amplified individually. And of course, like with any PCR process, you are introducing errors in your process. So the data or the reads will contain statistical or systematic errors here. And we then collapse again, and this we do analytically, we collapse the reads that belong to a specific strand of a specific molecule, and we create consensus reads. And we can then, on top of that, create a consensus read from the two reads that represent the individual strands of the DNA molecule. And if you're looking at the error rate that you can achieve or that you have to that you're encountering in your data, the second column in my diagram basically represents the regular data that you have, that you obtain from NGS sequencing, and a typical error rate in NGS sequencing is probably 1 in 100 to down to 5 in 1,000. When you apply simple molecular barcoding without regard for the strand that barcodes stem from, then you can achieve an error rate down to 1 in 10,000. And by applying this last step, by creating consensus read across strands, you can actually achieve an error rate as low as 1 in 10 million. And this is noise as low as you need it for liquid biopsy. I'd like to give you an example how this looks like using numbers from a real sample from Simon. His NGS panel had a target region of only 14 kilobases. Every single sample was sequenced with 14.7 million reads, as an example. And in the first step, quality filtering is applied. So we remove all reads that are of lower quality or that do not that are not paired, or that where the pairs are not on the same chromosome. This only filtered out about 1% of the reads, and from these high quality reads we generated those consensus reads that I mentioned on my previous slide. And this basically boils down the data to mere 400,000 reads per sample. But these 400,000 reads are these highly reliable consensus reads that are basically free from noise. But you have to keep in mind that you need to start from this large number of reads. You need to have this large sequencing capacity available to obtain these high quality reads that allow you to call very rare variants. This is an example of a QC report from inside our software varvis. It's listing the 6 genes that were contained in the panel. And basically the important column is the column here indicated with a red rectangle, 500x. The numbers in this column indicate the percent of bases that are covered with at least 500x of coverage in this sample. And as you can see, in for almost all genes, we achieve well above 100%. And in general, this sample exhibited a raw coverage of 40,000 x, and after consensus building, we achieved an effective coverage on average of 1,400 x. This can be nicely visualized in the software. This is a graph showing the percent of bases covered at a specific coverage, and these graphs show that the coverage is very homogeneous. This means you are sequencing very efficiently, and you're not wasting reads in specific regions, and you're not missing coverage in others. Now let me talk a little bit about how can you actually validate the performance of your assay. We face several challenges with this specific assay. Well, of course, there's a benefit of the small target region, but the target region, a small target region, can be a challenge in the validation because you will find very few reference samples that actually contain variants in those target regions. So you have to be very creative and sourcing reference samples from different sources. And the best available guideline how to proceed here is still the guideline from the AMP and from 2017, Jennings et al. This guideline was written at the time with solid tumor analysis in mind, but the principles that are laid out there can also be applied to liquid biopsy. If you want to get an idea how you can extend this, the guideline also to liquid biopsy samples, there is a nice paper from Clark et al. From Foundation Medicine. They are describing in detail how they are performing validation at scale using clinical samples. In our case, we were using various sets of reference samples, and we determined our limit of detection to be 0.2% in terms of allele frequency. And this is kind of a limit where we feel very safe. And I'm going to show you the details of the data that we obtained. The first validation data set was derived from genome in a bottle samples. Those are cell lines that can be obtained, for example, from the Coriell Institute. Those are, of course, they contain germline variants, and you generate or you simulate basically tumor variants by mixing those DNA samples. The important benefit that you have when using genome in a bottle is that the background is very well defined. That means you can very clearly determine the number of false positive variant calls in your samples. Kind of the drawback in terms of our small region of interest was that we could only achieve very few variants in the allele frequency range below 1%. But we were able to confirm all variants in those samples. We had no false negative samples. A second set of samples were obtained from the company Horizon. There, the spike-in is already included, but the drawback in those specific samples that were suitable for our assay was that the background was not well characterized. It was only characterized by exome sequencing, not to a level of quality that we needed in our case. So we were not able to make any statement about false positives in those samples. Again, we did not miss any of those variants. So far, we have no indication that that we're lacking any amount of sensitivity. There is another manufacturer of reference materials, SenseID, which provide ready-to-use spike-in and well-characterized background with a larger number of variants that can be used in our case. And we just haven't had the opportunity to characterize those samples, but we are very eager to proceed and extend the validation data. So in the end, we can say that sensitivity is very high. Well, since we have only so few variants in this allele frequency range below 1%, we are cautious, really specifying a number here, but we are very sure about a very high precision, close to 100%. We have not found any false positive variants, even though we were investigating across the entire assay. So the confidence interval is already very narrow here, but we are proceeding and still performing validation sampling. Well, let me at the end give anybody who is considering to establish a new assay with such challenging technology some specific advice. Consider the analytical performance very early on. There are various external factors that impact your assay design. First of all, the clinical application. What are the genes that you actually need to look at? What is the limit of detection that you need to achieve in order to make a clinically significant statement at the end? And of course, there will always be a certain budget associated with your assay. The sequencing costs must be reasonable. And those factors determine what is the actual genomic region of interest. So you need, make sure that you that you understand what the minimum region of interest is to make this assay useful. the limit of detections that you would like to achieve basically determines the technologies, also the technologies in the wet lab that you need to apply. And of course, you know, the budget that you have and the technology that you're applying determines what kind of depth of sequencing do you need to apply. And ideally, you're using a software that provides you specific feedback, how your assay performs, and that also, you know, saves you a lot of time by performing the validation procedure in an automated fashion. Okay, thank you. Yvonne?
Yes. I'm back. Thank you, Ben. So we can maybe continue to Q&A directly. I have some questions here. Maybe Peter wants to join as well. Good. Yeah, so there are you. Hi again. So I have a question regarding the validation. For Ben, can I also use clinical samples for validation?
Yes, you can. And as I mentioned, the paper from Foundation Medicine, they are detailing how they were using clinical samples. There are several things to consider with clinical samples, however. Those samples must be characterized to a high degree. And you also have to keep in mind, well, if you characterize those samples using next generation sequencing, then the probability to find any variants that you haven't found before, well, is very low. So you're comparing very similar technologies. Make sure that you also have reference samples in your validation set that challenge your own technology that have been characterized with other technologies as well.
Okay, I hope this answers the question in detail. By the way, I've seen that somebody raised his hand to ask a question. Please, if you want to ask another question, type in the question in the control panel on the right, so you can, because I cannot unmute you here. Next question. Can I use an NGS panel of 600 kb for liquid biopsy? Who wants to take this question, Ben?
Well, yeah, okay. So from a technical or from a bioinformatics point of view, well, of course, well, since to some degree the size is irrelevant, you know, from a data processing point of view. But of course, you know, it impacts kind of the cost factor. Larger region of interest means that you need to invest more money into sequencing capacity to achieve the required depth of coverage. So putting a lot of thought into those calculations in the beginning really pays off in the end, because you're not wasting time and money, you know, designing too large of a panel, and it's really makes sense to focus on really a region of interests that are important.
Okay. Next question for Simon. Does the panel capture the whole coding sequence of the 5 genes, or only focused on hotspots?
It's designed for the whole coding sequence for all 5 genes.
All included. Okay.
Yeah.
And how many samples can be sequenced on a mini-seq run?
Oh, for the validation runs, we only run 4 patients per flow cell per run. So yeah, this is, yeah, in terms of sequencing cost, this is not the ideal platform, but yeah, we use the mini-seq for the validation, and of course it would be better to use another platform like the NextSeq or even higher.
Okay. There are more questions coming in. How many redesigns were necessary to achieve the level of quality that you have presented here?
This was the first design for this panel. Yeah. Yeah.
One question for Peter. How do you deal with the fact that the IDT technology can detect sequencing from 100 base pairs length upwards, but not shorter ones? There's evidence that ctDNA can be found more often in shorter fragment part of plasma DNA?
Okay, I think the question is how probes that have a length of 120 base pairs would be able to hybridize to shorter fragments. Well, that is just exemplified by the fact that it can happen. So yeah, cfDNA can be short, but the probes will hybridize to the shorter fragments and fish them out of the libraries. And the sequencing then starts from the probes are eluted from the captured molecules, and the sequencing is starting from the adapters that have been ligated on into the during the library prep. I hope that answers the question.
And we see that in the data, of course. So with liquid biopsy samples, we see, for example, if you're sequencing at 150 cycles, we see that we are sequencing into the opposite adapter, but that's a bit bioinformatics task to take care of that.
Okay, thanks for the clarification, Seth. So thank you. Next question. Can the software detect the exact area of the affected sequence as mentioned in the slide before? Please clarify.
The affected area. Maybe can we go back a slide? Or yeah, I didn't quite understand.
I'm not sure if the question is. Can you specify the question? Can the software detect the exact area of the affected sequence?
So I mean, mostly, at least in the validation data, we were dealing with single nucleotide variants and only a few more complex variants. So the bioinformatics part, the variant calling is very straightforward here. It is simply that we can really, I mean, the library preparation and the addition of molecular identifiers allows us really to trace back the variants that we find to individual molecules. And when we find variants in more than one molecule and two separate molecules, we are obviously very sure that the variants exist in the sample.
The question was about the cancer cell affecting the sequence. Does this answer your question? I'm sorry.
But we are happy if, if we didn't get the question correct this time, please follow up with us by email. We are happy to enter into a discussion and clarify any questions that remain.
Okay. We will come back to you by email. Okay. There's another question to Peter. Are your probes ssRNA, ssDNA, or dsDNA?
Our probes are single-stranded DNA probes of 120 nucleotides, biotinylated.
Okay, I think all questions are answered. Yes, you will get a recording of the session afterwards. We will send a follow-up email to all participants. With this, I would like to conclude. I thank you all for your attention and our speakers for presenting here today. And I hope to see you again in the next session of the varvis webinar series coming up soon. Bye for now.
Bye-bye.
Have a nice day.
Bye.
Bye, and thank you.